Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. Now, we will go to lecture 24, where the role of elicitor in modulating alkaloid accumulation, particularly indole alkaloid accumulation will be covered. So, let us go to the concepts covered. So, before I go to the elicitor treatment for uplifting of indole alkaloids, particularly on the mechanism aspects, I will go for a quick revision whatever I have covered in the earlier class. Okay. So, let us go to the board now. What we have covered in the last class was the early steps of indole alkaloid biosynthesis. So, there what we have seen that tryptamine and secologanin. So, secologanin is basically coming from the terpenoid pathway. So, from geraniol through multiple steps, and this tryptamine and secologanin they joins together and form the strictosidin, and this strictosidin subsequently subsequently forms strictosidin aglycone by the action of glucosidase. So, and the enzymes responsible are, I have already mentioned, this one is TDC, and actually these two joins together. So, I must show it in that way, otherwise again you will be confused. So, sorry. And actually tryptamine originates from tryptophan, and now we put the enzyme. So, this was tryptophan decarboxylase, this one strictosidin synthase, and this one this strictosidin glucosidase, which makes strictosidin aglycone, and strictosidin aglycone is a very active compound, and that leads to formation of more than 300 different indole alkaloids in different plants. Now, but from strictosidin aglycone there are 3 different skeletal structures originate, and that leads to different group of alkaloids. So, one major alkaloid what I have mentioned in the previous class, that was azmalicin, another one is catharanthine, and another one is vindolin. Now, the structures these 3 alkaloids, the chemical structure, the structure is different, the skeletal structure. So, this was this comes under corinanthine type, corinanthine type. In one of the later classes, I will show you the structure in the form of slides. This one is called ibogan type, and this one is called plumeron type. What I have also shown that from this strictosidin aglycone, it forms 421 dehydro gaseocyzine. So, big name, and from dehydrogaseocyzine different routes originate. One route goes towards catharanthine and vindoline. This we will study in detail, and the other route which goes towards cathenamine and cathenamine can be converted into azumylicin or iminium cathenamine and this subsequently formed tetrahydroalstonine. So, this name I have not mentioned yesterday. So, this is tetrahydroalstonine, okay, and azumylicin can be converted into serpentine. Now, tetrahydroalstonine actually one can find in the plant called Alstonia. Alstonia species, different species are there. So, there this alkaloid accumulates. So, this is in brief. I have made the revision for you, and this is tetrahydroalstonine synthase, one enzyme is working here, and this conversion is by peroxidase, right. So, this is a brief revision. So, this is more or less I have covered. So, now we go to the next aspect, that is the mechanism of elicitor induced upliftment of this alkaloid. Okay. So, if you remember that I have drawn this diagram in the previous class, where it is showing basically a model for elicitor signal transduction leading to strictocytin synthase gene expression. So, what we see here, that in the membrane there is a receptor and the elicitor molecule comes and bind, and that leads to influx of calcium channels, which can directly without the involvement of jasmonate can activate a transcription factor like CRBF, okay. And that basically binds in the elicitor response elements, which is also called BA, and the other one is that elicitor, when it attached to that in turn also activates the octadecanoate pathway leading to the synthesis of jasmonates, and that jasmonate will be perceived by the jasmonate receptor, and then through a protein kinase activity ultimately this activates one transcription factor like ORCA, which comes and joins. So, now this aspect we will see in detail in the next slide. Then in the board. So, this is about the mechanism of activation of indole alkaloid pathway in say Catharanthus roseus upon yeast extract treatment. So, let us draw the diagram first. So, this is what is the plasma membrane, where there must be a receptor is basically a protein. So, this is what we can say receptor, and this is the elicitor, that is yeast extract. It basically comes and bind here, okay. And so, as a result of that, what is going to happen? Let us see in a very brief way. This is what is there inside the nucleus. So, this is nucleus, this is cytoplasm, and this is the plasma membrane. So, what happens that receptor upon binding with the yeast extract, it activates the jasmonic acid. So, that means leading to the activation of jasmonate. So, one arrow we can put it here and write it JA. JA stands for jasmonic acid, and increased level of JA I put it in arrow upper side, that means it is increased accumulation of jasmonic acid level. Okay, and that once it increase, let us see what is going to happen, but before that I need to draw here in the inside the nucleus. Okay, so, one is for this is a part of the promoter. So, this is an element, upstream element, and then you have the coding region for TDC and so on. Okay. So, we will have to also draw the STR. Well, then, so this is the promoter region, here lies the Tata box for STR, and this is nucleus. Next, what we see, this jasmonic acid, what it does, it activates the ORCA3. So, activation of ORCA3 which comes and bind near the data box. So, this is I will put ORCA, not 3, I am not mentioning here, ORCA, octadecanoid responsive catharanthus apetal elements. So, it is a positive regulator of this TDC biosynthesis, and not only that, this yeast extract also activates another transcription factor which comes and bind here, which is called BPF1. Now, these are the positive regulators which will allow the STR gene to express and TDC gene to express. Okay. And ORCA3 also activates the STR as well, but there are some transcription factors which are basically are the negative regulators. For example, like ZCT, and like I put it in a different color, like ZCT, and GBF. So, these try to block the activity. Now, ZCT is basically a zinc finger catharanthus transcription factor, and BPF1 is basically box P binding factor. It is actually belongs to this BZIP class, and GBF is basically the G box binding factor. So, what happens in normal cases, this ZCT and GBF it binds. So, and it binds in the promoter region, and that is why it is not allowing the STR or TDC gene to be transcribed. So, upon the yeast elicitation happens, so that activates the jasmonic acid, and through jasmonic acid ORCA3 is getting activated. Otherwise, without this also, okay, it also activates directly the BPF1. So, these are the positive regulators, and once these are activated, so that actually remove the negative regulator and it binds, and as a result of that what we see the transcription of these genes, that means transcription of the STR as well as TDC. This is in brief mechanism of activation of indole alkaloid pathway in Catharanthus roseus culture upon yeast extract treatment, Catharanthus roseus culture. So, it could be hairy root culture, it could be shoot culture, it could be cell culture. So, next we will see another model in the next slide. This model I briefly mentioned in one of the earlier classes when I have spoken about this elicitation in general, but we will again discuss this because this is directly in this relevant to this context. So, this is a model for jasmonic acid, J stands for jasmonic acid induced STR gene expression. STR means strictocytin synthase mediated gene expression mediated by what we can say mediated by the mediated by, okay, I will put the not here, mediated by the OR CA3 transcription factor in Catharanthus roseus culture. So, jasmonate, okay, I put the jasmonate towards little bit left, otherwise I need the board a bit more. So, go to pen and take this color. Jasmonate activates jasmonic acid receptor, and that by some unknown mechanism activates a general transcription factor which was not fully known yet. So, that is I put it in TAF. TAF is basically transcription activating factor. TAF, I will put it TAF here. TAF stands for transcription activating factor. So, that is getting activated. So, TAF, when it is activated, how I will denote? I will denote by adding a star. So, this means that it is now activated, and then which in turn will activate the specific transcription factor required for activating the pathway. That specific transcription factor is basically the ORCA, and 3 is one specific one. So, this ORCA3. So, ORCA3 will get activated size is not good. So, activation means I put this star means activated, and this activation what is going to happen. So, there are two things. One, let us draw the DNA. So, here lies a jasmonate response element. JRE stands for jasmonate response element, TAF stands for transcription activating factor. Jasmonate response element. So, here this TAF, activated TRF, will come and bind. So, I have to put some arrow to add it here. So, like this, and that basically what will happen, it will activate the genes for ORCA3. So, that leads to activation of ORCA3. So, what we put mRNA. So, this mRNA is basically showing activation of ORCA3. So, maybe ORCA3 I put it in this color. So, so this is activates, and then what will happen, that it leads to activation of ORCA3, and so that means and then ORCA3 is getting activated. So, this ORCA3 will come and bind here. So, where is the ORCA3? Here is the ORCA3, it will come and bind ORCA3. So, the activated ORCA3 will come and bind. So, it is activated, and where it binds? It binds in the DNA where lies the jasmonate and elicitor response element. So, JERE stands for jasmonate elicitor response element. Okay, and once it is there, then what it does? It basically activates the STR gene. So, this is mRNA. So, what I will write here? This leads to activation of STR gene. So, activation of STR gene, and that leads to the formation of the STR protein, and which plays important role in binding tryptamine and cycloguanine, leading to the formation of streptozidin. So, this part is basically the ORCA3 activation, ORCA3 activation, and this is all is basically this transcriptional cascade. So, this is in brief a model for jasmonate induced STR gene expression. So, what STR does is basically STR converts tryptamine and cycloguanine tryptamine. Cycloguanine to streptozidin, and pathway moves. So, the STR plays very important role. So, here is the STR. Okay. So, this is all about the model for jasmonate induced STR gene expression. Now, with this I will end this class. I will not go into the specific examples of hairy root culture expressing the transcription factor. Maybe in one of the subsequent classes I will bring that issue. So, with this I will end this class. Thank you.